Okay, so I can see that people keep joining us, so I think we can start. Good afternoon, everyone. My name is Anna Yanaeva, and I'm Head of Business Development at OCT Clinical, a full-service European CRO. And I'm glad to introduce to you uh, Georgy Smirnov, who will be our speaker today on our webinar on demystifying the intricacies of analysis datasets and ADaMStandard. Georgy is the head of statistical programming at OCT, also acting as a senior SAS programmer. And today, Georgy is going to share part of his impressive experience, more than 20 years experience in the field. Hello, Georgy. How are you today?
Thank you, Anna. I'm fine.
Good. So before we move on, just a couple of tactical moments for all the attendees. On the right side of your screen, you can see a chat box, and you can type your questions there throughout the webinar, and we'll be taking all the questions at the end of the event. So, well, now, Georgy, the floor is yours.
Thank you again, Anna, for the introduction. Good afternoon, everyone. And let's start. At the next slide, you can see the agenda items we are going to cover. What is an analysis dataset? ADaM in the CDISC quote. Why does FDA want ADaM? Goals of ADaMStandard. Fundamental principles of ADaMStandard. Implementation strategies for ADaMStandard. Types of ADaM datasets, which are subject level analysis dataset or BDS, basic data structure or ADSL, basic data structure or BDS, and occurrence data structure. And the last one is summary of the most recent updates to ADaM standards. So, what is this analysis dataset? In general, analysis datasets or analysis files are the data used for statistical analysis and reporting. An analysis dataset serves as a central depository of raw data and analyzable variables derived from one or more of the raw datasets. The derived variables from analysis datasets are used as inputs to produce statistical summaries, such as tables, figures, and listings supporting a statistical analysis by means of SAS programming. Although it is technically possible to produce tables, figures, and listings directly from raw data, using the analysis datasets can save programming time, increase productivity, and facilitate quality control procedures. So it is the best practice. The following flowchart describes the process. Analysis datasets are derived based on raw data using the methodology described in statistical analysis plan, and then used as inputs for producing of tables, figures, and listings. The next item is ADaM in the CDISC world. ADaM is abbreviation for Analysis Datasets Model suggested by the Clinical Data Interchange Standards Consortium, or CDISC. CDISC defines and manages industry-level data standards that are widely used during the analysis, reporting, and the regulatory submission of clinical data. For instance, the Study Data Tabulation Model, or SDTM, is a submission data standard into which raw study data are mapped and collated. ADaM is a companion standard for use with analysis data, and it is the best practice to use SDTM data as a source for these datasets. Doing this allows for the easy documentation of any data processing with defined XML, which is the CDISC standard for data definition files. This presentation is based on the following documents which define the ADaM standard. The Analysis Data Model version 2.1, which defines the underlying fundamental principles. The ADaM Implementation Guide version 1.2, which describes standards implementation for analysis issues as well as variable names and labels. And the Implementation Guide Appendices, which recommend structure for specific analysis issues, such as Basic Data Structure for Time-to-Event Analysis, version 1.0, and Structure for Occurrence Data, version 1.0, for adverse events, concomitant medications, and medical history analysis datasets. You can also see the publication date of these documents in the slide. These are also some recent updates to the standard documents. We will briefly discuss them at the end of this presentation. The very important point is that US Food and Drug Administration, or FDA, requires ADaM datasets to be included into submissions. Why FDA? Why does FDA want ADaM? Standards are developed and used for many reasons, including to increase efficiency. The US Food and Drug Administration Study Data Technical Conformance Guide states that ADaM facilitates their review, simplifies programming steps necessary for performing analysis, and promotes traceability from analysis results to ADaM datasets and SDTM datasets. One of the expected benefits of analysis datasets that conform to ADaM is that they simplify the programming steps necessary for performing an analysis. As noted above, ADaM datasets should be derived from the data contained in the SDTM datasets. These are features built into the ADaMStandard that promote traceability from analysis results to ADaM datasets and from ADaM datasets to SDTM datasets. This implies the main goals of ADaM, namely quality, support clear and ambiguous communication of statistical aspects through structure and content of data, traceability, allow better understanding of data derivation sources, efficiency, allow more efficient generation and review of analysis results by providing data sets that are ready to use for analysis. Metadata, enable documentation of structure and content by providing metadata on datasets, variables, parameters, and results, but also on methodology and statistics. And submission, standardization of analysis datasets to meet the requirements of the FDA. To meet these goals, the four fundamental principles have been introduced. Be analysis ready, have metadata, support traceability, be usable with commonly available tools. Let's discuss these principles in more detail. The first principle, ADaM requires the datasets to be analysis ready, which allows the application of analysis from ADaM datasets with little or no further data manipulation. Ideally, there should be one statistical procedure away from the analysis dataset to the statistical results, one SASPROC away principle. It also eliminates or greatly reduces the amount of programming required by the FDA or other regulatory agency reviewers. Please note that one PROC away principle doesn't mean that a formatted table can be generated in a single statistical procedure. Rather, it implies that each statistic in the table can be replicated by running a standard SAS routine using the analysis dataset as input. Please also note that one analysis dataset can be used to generate more than one analysis result. But in this case, some supportive variables should be in place to facilitate easy selection of data. Supportive variables such as selection criteria or flags should be specific and sufficient and easily be included in a where clause or something equivalent to ensure the exact set of records, to ensure selecting the exact set of records appropriate for a given analysis. At the slide, you can see an example of such a clause which allows to select on-treatment observations related to the second phase of the study. This is just an example. The second principle, ADaM requires to have metadata describing the analysis. Metadata is the method to allow clear and concise communication of analytic results to the sponsor and to the regulatory reviewers, which includes statistical methods, transformations, assumptions, derivations, and imputations performed. Metadata for analysis datasets has four sources: statistical analysis plan for description of statistical methods, assumptions, and rules for imputations and derivations; analysis dataset specification document for transformations, specifications of algorithms, and dataset and variable information, for example, code list used; SAS datasets themselves for actual information of the data, for example, code used, final variable lengths, etc., and analysis data reviewers guide or ADRG document. All the above information is linked together in the defined XML file. The third fundamental principle is traceability. ADaM supports traceability by providing metadata traceability for datasets, and data point traceability. Metadata traceability is provided by the analysis dataset specification document and/or define XML file and enables the user to understand the relationship of the analysis variable to its source datasets and variables. This relationship is given by description of algorithms or references to statistical analysis plan for algorithms or by combination of both methods. Data point traceability is facilitated by having variables in the analysis dataset only for the sake of traceability of values back to the SDTM variables. Let's look at the example of ADVS, vital signs analysis dataset at the slide. Analysis variables here are parameter, analysis value, analysis visit, and its numeric representation. SDTM variables are highlighted here. All of them must be 100% identical to source SDTM domain. They are included into analysis dataset for traceability purposes. The fourth principle, ADaM datasets have to be usable with commonly available tools. It restricts the file transfer to SAS version 5 transport files for data, because this is the last open format usable without a SAS license. To be compatible with this format, variable name should not be longer than 8 characters and not starting with underscore. Variable label should not exceed 40 characters, and maximum text field length is 200. Okay, now we move on to implementation strategies for ADaMStandard. The general rules here are as follows. Create with specific analysis in mind. Create the optimal number of analysis datasets. ADaMStandard requires at a minimum the analysis dataset for subject level data, or ADSL, to be created. Analysis dataset can be dependent on each other. However, this should be avoided whenever possible to allow independent updates of single datasets, except dependencies to ADSL dataset. The optimal number of ADaM datasets for the purposes of specific analysis can be chosen based on the following points. One analysis dataset can be used to generate more than one analysis result, as mentioned earlier. Data used only for listings, not for analytical displays such as tables or figures, do not need to be collated into analysis-ready datasets. There is no requirement that every SDTM domain has a corresponding analysis dataset. If sponsor requirements do not say otherwise, create enough datasets to allow all tables and figures to be produced from the ADaM, and also include all SDTM records to the ADaM, where possible, to allow listings to be produced directly from the ADaM as well. Use only SDTM data as a source for ADaM and TFL programming. Direct references to raw data are generally not allowed. Use of external data should be avoided as far as possible. The ADaM naming conventions are to be followed during implementation. Subject level analysis dataset should be named ADSL. All other dataset names should start with prefix AD. ADaM does not have control terminology for dataset names other than ADSL. However, try to make dataset names informative. For example, ADF for efficacy, ADQS for questionnaires, ADAE for adverse event data, etc. Try to be consistent with SDTM domain used as the data source. For example, ADAG, ADVS, if a single SDTM domain is used as a source, like AG or VS. For standard ADaM variables, the naming convention should follow the standardized variable names defined in the ADaM implementation guide. Any variables copied directly from SDTM data into an ADaM dataset shall be used unchanged, with no change made either to the attributes, such as name, label, type, length, etc., or their contents. Same meaning, same name, same values principle. Sponsor-defined variable names can be given to any other analysis variable that is not defined within the ADaM or SDTM standard. Please also note that user-defined formatting of numeric values is not allowed. Instead, define a pair of variables, numeric and character, with code list. Okay, let's move to types of ADaM datasets. While SDTM domain classes are determined according to data type, such as interventions, events, or findings, the ADaM equivalents are classified by analysis approach. There are three standard structural classes of ADaM datasets: subject-level analysis dataset, basic data structure, and occurrence data structure. Subject-level analysis dataset, or ADSL. Let's consider some key points. This dataset contains one record per subject, regardless of study design. It contains key information about each subject. It allows for easy merge with any other dataset, for example, merging all population flags and treatment variables. It is the only dataset that must be created for a study being submitted in CDISC standards to the FDA. It is the source for subject-level variables in other ADaM datasets, such as statistical model covariates, population flags, subgrouping variables, etc., needed to enable analysis of the PDS dataset. It is also used to obtain denominators for percentages calculated based on whole populations. It is technically feasible to take every single data value in a study and include them all as variables in a subject-level dataset, such as ADSL, but that is not the intent or the purpose of ADSL. ADSL is used to provide key facts about the subjects that are analysis enabling or facilitate interpretation of analysis. ADSL is not the correct location for key endpoints and data that vary over time during the course of the study. It should be noted that though ADSL is a source for subject-level variables used in other datasets, there is no requirement that every ADSL variable should be copied into a BDS dataset. Other subject-level variables that are important in describing subject experience in the trial are also included in ADSL. At this slide, you can see some examples of variables that are required to be included into ADSL dataset with the corresponding labels. Study and subject identifiers, study identifier, subject identifier for the study site identifier, and unique subject identifier. Subject demographics are age unit, sex, and race. Population flags. At least one population flag is required. For example, safety population flag and PK population flag. Treatment variables are required if there is an investigational product. Date of first exposure to treatment and date of last exposure to treatment. Here are some examples of additional ADSL variables. Baseline characteristics such as height at baseline and weight at baseline, and stratification variables or protocol-specific event information, date of these, and a COG score at baseline. At this example, you can see how these variables should look like. Please note that the population flags should have values of Y for subjects included into specific population, or N for subjects excluded from it. Null values are not allowed for population flags. Some other points regarding population flags. Population flag is required in ADSL for every population that is defined in the statistical analysis plan. A minimum of one subject-level population flag variable is required for every clinical trial. The values of subject-level population flags cannot be blank. If a flag is used, the corresponding numeric version with the suffix fn, where 0 equals to no and 1 equals to yes, can also be included. Additional population flags may be added. The below list is not meant to be all-inclusive. Full analysis set population, safety population, intent to treat population, per protocol population, completers population, randomized population, and enrolled population. Other treatment variables to be included into ADSL are as follows. ARM, mapped from SDTM DM ARM variable. TRTXXP is a subject level identifier that represents the planned treatment for period XX. In a one-period randomized trial, TRT01P would be the treatment to which the subject was randomized. It might be derived from ARM variable from SDTM DM domain. TRT01P is required in ADSL. TRTXXA is a subject level identifier that represents the actual treatment for the subject for period XX. It is required when actual treatment does not match planned, and there is an analysis of the data as treated. Planned sequence of treatments is required when there is a sequence of treatments that are analyzed, for example, in a crossover design. Actual sequence of treatments is required if a situation occurred in the conduct of the trial where a subject received a sequence of treatments other than what was planned. Planned pooled treatment Y for period XX. Useful when planned treatments in the specified period XX are pooled together for analysis according to pooling algorithm Y. And actual pooled treatment Y for period XX. Required when the corresponding planned pooled treatment is present and actual treatment for this period is also present. Okay. At this slide, there is a snapshot of ADSL dataset containing the required treatment variables. As you can see, subject 1044 received actual treatment that did not match planned. So TRT01A is a required variable. If there is no such situation in the trial, TRT01A could also be included, but is not required. Let's move to the basic data structure. The basic data structure is another main class of ADaM datasets. It contains one or more records per subject, per analysis time parameter, and per analysis visit or analysis time point. BDS has analysis-focused design. Analysis parameter and analysis value are key variables in the structure. It includes both observed and derived values required for the analysis. It is possible to add derived analysis parameters if required for an analysis as well. An example would be where a derivation uses results from a number of different parameters, or where a mean is calculated at subject level from all the values collected for a subject. Derived records also may be added to support last observation carried forward or worst observation carried forward analysis. The basic data structure has been defined by ADaM to be flexible enough to support most methods of analysis used in clinical trials, such as analysis of variance, analysis of covariance, categorical data analysis, logistic regression, CMH test, Wilcoxon rank sum test, time to event analysis. BDS also improves the quality and efficiency by providing a predictable data structure. It is easier to move between projects, allows reuse of programming code, and consistently aids creators and users of analysis datasets. At this slide, the main types of ADaM BDS variables are present. Identifier variables. Basically, they are the same as for ADSL, except analysis sequence number. Analysis parameter variables. Timing variables. Here, analysis relative data, or ADT, is usually calculated from the data of the first study drug intake. Indicator variables. Population variables, they usually should be merged directly from ADSL. analysis descriptor variables, time to event analysis descriptor variables, predictor and covariate variables, which are also merged from ADSL, data point traceability variables, and DTM variables, also included for traceability purposes. At this slide, you can see the visualizing of the basic data structure, so you can imagine how these typical BDS variables could look like. This is a snapshot of ADAG, AD, ECG analysis dataset. Here, analysis parameter is QTCF interval measured in milliseconds. Please note that the unit of measure is also included into param variable. Analysis value is 482. Baseline value for the circuit is 446. And change from baseline is 36. The observation is taken from the source EG domain. Therefore, derivation type here is null, and some SDTM variables are also included for traceability. Since the analysis value is higher than the upper limit of normal, the analysis reference range indicator is set to H. Predictor and covariate variables and population flags are merged directly from ADSL dataset. Let's discuss analysis parameter variables in more detail. Parameter is a required variable in BDS. It includes test units, specimen types, locations, positions of measure, etc. as appropriate. There is no need to include additional qualifier variables. It may be longer than 40 characters in length, but no longer than 200. Parameter code, or param-cd, is also a required variable. It represents short name or abbreviation of the analysis parameter, no longer than 8 characters. It must be a one-to-one mapping with parameter. Permitted analysis parameter variables are param-n, the numeric code for analysis parameter, and parameter category y, categorization of analysis parameters. Y here is a substitution for an integer number like 1, 2, etc. In this example, analysis parameter values param, param-cd, and param-n are framed. This is a snapshot of laboratory analysis dataset, ADLB. Please note that a new parameter, LDL to HDL ratio, has been derived for analysis purposes within the analysis datasets, not taken from the source data. In such a case, a variable param type could be added. Param type value for these parameters is derived to highlight the fact that the whole parameter is derived within the analysis dataset. Consider BDS analysis variables. Aval, or analysis value, is a numeric analysis value described by parameter, and aval C, or analysis value C, is a character analysis value described by parameter. At least one of aval or aval C variables is required in BDS. There must be a one-to-one mapping between aval and aval C within one parameter value if both variables are filled. Base represents a numeric baseline value. It is required if dataset supports baseline analysis. And base C is a character baseline value. It must be one-to-one map between base and base C, similar to aval and aval C. CHG is a change from baseline. It equals to aval minus base and should be populated for all post-baseline records of param. Percent change from baseline could also be included if needed. Analysis category Y, where Y substitutes an integer number like 1, 2, etc. as before, is a categorical representation of aval and or aval C. A one-to-one mapping to aval and or aval C is not necessary here. And analysis category YN is a numeric representation of analysis category Y. There are other variables similar to the analysis category variables, which include baseline, change from baseline, and percent change from baseline categorization. At this slide, you can see the snapshot of ADLB dataset as an example of how some of these variables could look like. The analysis value here is a LDL level. The first and second rows correspond to pre-baseline and baseline measurements, respectively. So the second row is marked with baseline flag equal to Y. The next example illustrates the case where the main variable for analysis is categorical. Our C variable contains text or responses to some question like good, normal, bad, very bad, and aval represents the numeric coding. Aval cat1 is further categorization of answers into groups like good, very good, or bad, very bad. Now let's look at treatment variables in BDS. TRTP is a record level identifier indicating the planned treatment for the corresponding period. It is different to TRTXXP in ADSL, which is a subject level variable. However, if populated, TRTP must match at least one value of the character planned treatment variables in ADSL, for example, TRT01P. In implementation guide version 1.0, TRTP was a required variable. From version 1.1, it has become a conditional variable. However, at least one treatment variable is still required in BDS. TRTA is similar to TRTP, but indicates the actual treatment. TRTPN and TRTAN represent numeric codes, which are a one-to-one mapping to the respective character treatment variables. Moving on to analysis descriptor variables, let's briefly discuss derivation type or dtype variable. Dtype is used when an observation is derived or imputed within a parameter. So analysis value and character analysis value are not obtained from SDTM data. Examples of when dtype is used are baseline derivation, last observation carried forward imputation, dtype should be equal to low CF in this case, worst case imputation, dtype should be WC. Control terminology for dtype can be found using the link at the slide. Please also note that dtype is not used if a whole parameter is derived. In this case, param type could be used instead, as mentioned before. The example at the next slide shows a case when average of two values is used as a baseline, but could also be a worst case, minimum or maximum, etc. The baseline value is the average of screening and J1 values. Analysis visit is set to baseline, numeric analysis visit is set to 0, derivation type is given value of average, and baseline record flag is set to Y. The next example illustrates last observation carried forward approach. If a subject has a missing scheduled visit, low CF imputation could be applied if specified in the statistical analysis plan. Double asterisk STRSN in the value from the source STM domain. For example, could be VS STRSN or LB STRSN. This variable is kept for illustration purposes. Last observation carried forward imputation is completed for missing values of the result at week 5, week 6, and week 8, with the type given value of low CF. The imputed records are highlighted at the slide. At the conclusion of this section, we consider BS implementation for time-to-event analysis. A variant of the BDS is available for TT analyses that are commonly used in therapeutic areas like oncology. This variant additionally contains a variable for the original date of risk used for the start times in any TT analysis or censoring for subject where the events of interest are not observed. In the context of TT analysis date is the event or censoring date for the subject. Analysis value is time to event itself. And analysis descriptor variables for TT are time to event origin start date and censor, which defines whether the observation was censored. It is strongly recommended to use 0 as an event indicator and positive integers for censoring indicators. And event or censoring description is the text or description of the event of interest or censoring reason. An example could be seen in the next slide. Please note that the ADaM dataset here contains the sub-event data used to derive the analysis parameter time to hypertension event. This composite endpoint is of primary interest here. Subject 2010 was admitted to hospital at day 9, so the analysis value for time to hypertension event is set to 9, and censor is given value of 0. Subject 3082 completed the study at day 10 without blood pressure increase and admission to hospital. Therefore, analysis value is 10 and censor is 1, as this is a censored observation. And the third structural class of ADaM datasets is occurrence data structure. Occurrence data structure is based on both ADaM Analysis Data Model version 2.1 and ADaM Implementation Guide version 1.2. This type of ADaM dataset is used for occurrence analysis, which is the counting of subject with a given record or term, and often includes a structured hierarchy of dictionary coding categories. It is used mostly for analysis of adverse events, concomitant medications, and medical history. The structure for the occurrence data is usually one record per each record in the corresponding SDTM domain, like AE, CM, or MH. Though some exceptions exist, for example, when the topic spans several treatment periods and needs to be counted in each of them. Thus, multiple records might be needed. Or adverse events need to be analyzed along multiple coding paths. Occurrence data structure has some specificities. It is mostly used for the comparison between treatment groups of data, summarizing the number of subjects with at least one occurrence of a term. So there is no need for eval or eval-C variables or analysis versions of the dictionary coding variables. The documentation for occurrence data should contain the name and the version of the dictionary used for coding, if any. Occurrence dataset must contain all variables from the SDTM dataset and the corresponding supplemental qualifiers that are needed for analysis or traceability. It might contain different levels of a coding hierarchy. Denominators usually need to be obtained from ADSL rather than directly from the occurrence analysis dataset, since some subjects in the population may not have any records in the corresponding SDTM domain. Dictionary coding might evolve during the trial, for example, from interim analysis to final analysis. Occurrence data variables are categorized as such: ADSL variables, for example, safety population flag, identifier variables, dictionary coding and categorization variables, timing variables, indicator variables, occurrence flag variables, treatment and dose variables, descriptive variables, and any other useful variables. Identifier variables are needed to uniquely identify a patient and an occurrence in an occurrence dataset. You can see them framed in this example. The only required identifiers are study identifier, unique subject identifier, and sequence number from the source domain. Here, double hyphen should be replaced by the SDTM source domain name, for example, AE. Analysis sequence number would be recommended for traceability purposes if the occurrence dataset is the source for another analysis dataset, for example, time to first severe adverse event in ADTT. Please note that such IDTT dataset should have basic data structure, not occurrence, as it is a time-to-event dataset. Dictionary coding and categorization variables are shown in this example, such as AE preferred term, AE system organ class, etc. Include in occurrence dataset all the appropriate coding or categorization variables that are required for analysis purposes. Analysis categories could also be created using variable acat_y if needed, acat_1 in this example. The metadata for each coding variable should include both the name and the version of the dictionary. Timing variables can be seen in the next example. Timing variables could be useful for traceability purposes, and some of them should thus be included from the corresponding SDTM domain. For example, start date time of adverse event. In addition to this, timing variables are often needed for analysis purposes, for example, for derivation of treatment emergent flux, or to assign an event to one of the study periods, or calculate the event's duration. If needed, imputation of partial or missing dates can be done in analysis datasets. In this case, appropriate imputation flag should be added. You can see this in the second, third, and fourth records. Here is analysis start date and analysis imputation date imputation flag, analysis start date imputation flag. Period and phases timing variables might also be included if analysis by period or by phase need to be performed. The example of such variables here is analysis phase. Indicator variables. Any indicator variables needed for traceability or analysis purposes should either be copied from SDTM or derived in the occurrence dataset. A treatment emergent flag is generally needed for event analysis and should be derived in occurrence dataset. The SDTM treatment emergent flag variable could also be included to highlight the differences between the analysis and the SDTM variables. Other analysis indicator variables such as pre-treatment, on-treatment, and follow-up flags could also be added if needed. Treatment and dose variables are shown in this example. This is a frame here. At least one treatment variable is required, such as TRTP, TRTA, TRTXXP, or TRTXXA, depending on the analysis to be performed. TRTP or TRTA would be derived from ADSL variables TRTXXP or TRTXXA. These are record-level variables similar to those in BDS datasets. Dose variable may also be added if needed for analysis purposes. For example, treatment dose, cumulative dose, unit, etc. Descriptive variables might be needed for analysis. For example, event severity for adverse events, dose routes for concomitant medications, etc. If a variable from SDTM domain needs to be re-derived for analysis purposes, then a new analysis variable needs to be derived, as the original SDTM variable has to be included in the analysis dataset for traceability. And this example, the variable AESIV is copied from the source SDTM domain, AE domain, and a derived variable ASIV, analysis severity, is added for analysis purpose, together with its numeric coding. Okay, and the last topic for today is a summary of the most recent updates to ADaMStandard. Recently, CDISC has released a new version of the standards for SDTM, as well as some updates to ADaMStandard. ADaM implementation guidance version 1.3, ADaM OCCDS implementation guidance version 1.1, ADaM implementation guidance for medical device version 1.0, and ADaM implementation guidance for non-compartmental analysis version 1.0. The new ADaM structure for occurrence data version 1.1 contains 3 additional examples. Analysis of adverse events that change over time. For example, when the sponsor has decided to track changes of severity, causality, or seriousness in and finding about adverse events, SDTM domain. Analysis of adverse events from multiple input domains. For example, when the individual occurrences of an event of interest are tracked in clinical events domain, but then analyzed together with other adverse events stored in adverse events SDTM domain as usual. And analysis of protocol deviation, putting the emphasis on the need to have strong metadata traceability when data point traceability is not feasible, especially when an ADaM dataset is derived from multiple sources, for example, SDTM and ADaM datasets. In addition to the three examples above, the new version of the OCCDS introduces some new indexed variables, for example, TREM where there are multiple periods analysis needs. Example 4, analysis of treatment emergent adverse events in a crossover interaction study, has been modified to show the use of these new variables. And finally, the new implementation guide version 1.3 documents do not contain any major updates nor any new standard variables, with many changes to or additional of text to clarify topics or guidance already available in previous versions of the implementation guide. All the above new implementation guides are also listed in the ADaM implementation guide. So we have covered all of the items for today. Thank you for your attention.
Thank you very much, Georgy, for this immersion to the topic from the basic principles of ADaM datasets to the latest updates in ADaM standards. So it was great. Thank you for sharing your extremely useful information, your experience. We are ready to take several questions. We still have some time, and I can see a lot of activity here in the chat. That is great. First of all, yes, we will be sharing the slides afterwards to all the attendees. The next question is, what is the minimum set of variables in the data set so it can be considered ADaM BDS?
The minimum set of variables for BDS consists of study identifier, unique subject identifier, parameter, parameter code, at least one of analysis value or character analysis value, eval or eval C variable, at least one treatment variable, for example, TRTP or TRT01P, and at least one population flag.
Perfect, thank you.
This is the minimum.
The next one is, should I make an ADaM dataset for each SDTM dataset I have, including DV, IE, and CO?
No, you shouldn't. If any particular domain is used solely for data listings, then it is definitely no need to produce the ADaM dataset based on this domain. However, if any analytical display such as table or figure is planned to be generated from this domain, it would be preferable to produce a corresponding ADaM dataset first.
Okay. I can see a couple of more questions. What are the key differences between SDTM test CD variables and ADaM param CD? And I think the next one here is, why can't one use just test CD instead of param CD?
The key difference here is that for SDTM XX test CD variables, additional qualifiers could be needed to fully describe a measure. For example, if a blood pressure is measured in both sitting and standing positions, in addition to VS test CD variable in VS domain, we also need a VS pause variable to know in what position a given measure was done. In ADaM BDS, there must be different values of param CD for different positions, different values of param CD, so that no additional qualifiers are needed to uniquely describe the measure. That is the key difference. However, in case when there are no additional qualifiers to XXS test CD in the source SDTM domain, then its value could be copied to param CD without modification. And probably it is the easiest way to populate param CD in such a case. So the key difference is presence or absence of additional qualifiers.
Okay, great. think I can see one more. Let's see if we can cover this. Does a particular TFL have to be generated with a unique ADaM domain, or maybe generate using multiple ADaM domains? So is just one domain has to be used, or multiple ADaM domains can be used?
Generally, I would say that a unique SDTM dataset should be used as a source, if it possible, if it is possible.
Mhm.
And the intention of ADaMStandard is to have unique ADaM dataset for each analysis output, such as table or figure. So the particular table or figure preferably should be produced using one single SDTM domain as a source.
Okay, perfect. I think that was the last question we can cover now. But please feel free to follow up and reach out to us. You can see the email here. So just reach out to us with any further inquiries or further discussions. We're always happy to help. So again, Georgy, thank you so much. And thanks everyone for joining us. And bye.
Thank you very much. Bye.
Thank you.